Hello dear students, welcome to MBM Science Digital Learning. In today's lecture, we are going to study about paper 301 syllabus, unit number 2. So let's start. This paper title is Microbial Systematics and Environmental Microbiology, which is described by Saurashtra University. In this episode 1, I am going to discuss unit number 2, Overview and its key terminologies. Unit number 2 is Prokaryotic Diversity. Prokaryotic. There are two words, prokaryotes and eukaryotes. Now, what do you mean by word prokaryote? Pro means primitive and karyote, that means nucleus. So the organism which are having very primitive type of nucleus. So naturally they will not be having very well developed cell organelles. So these types of organisms are known as prokaryotes. Now, what do you mean by word prokaryotic diversity? Prokaryotic diversity means all those organisms who belongs to prokaryota. How they are classified, how they are found, they are classified and diversified. We'll be studying in this particular topic. So this unit deals with 4 subpoints. First is point number 2.1, that is Introduction to Archaea and Eubacteria. Now here we will be studying in detail about Archaebacteria and Eubacteria, their differences, which organisms are included in Archaea and Eubacteria. Next, we will be studying about the gram-negative bacteria. Now, we all are knowing that Hans Christian Gram gave two categories of bacteria. One, that is gram-negative bacteria, and second, that is gram-positive bacteria. Already you might have studied this in your previous semesters. So again, I request you all to brush up with the previous year syllabus so it will be easier for you to understand these topics in a better manner. Now, what all things we are going to study in this 2.2, that is gram-negative bacteria, general features of aerobic, microaerophilic, motile, that is moving, helical, that means helix-shaped, and vibrioid, that is coma-shaped. Non-motile, curved bacteria, then aerobic bacteria, microaerophilic rods and cocci bacteria, facultative anaerobes, rods, curved and helical bacteria. These all are categories of various types of bacteria. Then dissimilatory sulfate reducers. What are that? We will be studying. Then anaerobic cocci and phototrophic bacteria. That means the bacteria which are capable of producing their own food. So these are the things which we will be covering up in this particular unit. Next is, as we studied about gram-negative bacteria, we will be discussing regarding gram-positive bacteria. Why they are gram-positive bacteria? They are forming endospores. So we will be studying endospore formers, rods and cocci, asporogenous rods, Mycobacteria and Actinomycetes. Next and last, we'll be studying about extremophilic microorganisms. Now, what do you mean by what extremophilic? Philic and phobic. Microbiology is such type of subject where we discuss about various terminologies, where we come across one typical type of word many times. So philic means loving and phobic means hater, or which is repulsing. So extremophilic, so the organisms which survive or live in extreme environments like extreme hotness, extreme cold, extreme temperature, extreme pressure. So such type of organisms are termed as extremophilic microorganisms, or in short, we will say that they are extremophiles. So in this particular unit of prokaryotic diversity, we will be studying about Archaea and Eubacteria, gram-negative bacteria, gram-positive bacteria, and extremophilic microorganisms. Now, let us understand few of the key terminologies so you will become more aware about the words which are used in the syllabus. Later on, we will be discussing each of them in detail in further lectures. So, to begin with, first is Archaea. What is Archaea? Archaea constitutes a domain of single-celled organisms. Single-celled means only one cell will be present in the entire body. These microorganisms lack nuclei. They do not possess nucleus and therefore they are termed as prokaryotes. Archaea were initially classified as bacteria, receiving the name archaebacteria. Many a times people will say archaebacteria, but no, actually they are not bacteria, they are considered to be only archaea. Despite this morphological similarity to the bacteria, archaea possess genes and several metabolic pathways that are more closely related to those of the eukaryotes. Now, why they are not considered as bacteria? Because some of their metabolic pathways or metabolism where they carry out their routine functions are related or similar to various types of eukaryotes. What are eukaryotes? Well defined cell organelles are there, well defined nucleus is there, notably for the enzymes involved in transcription and translation. Now, transcription and translation you might have studied in your school days, right? That is the central dogma of the life. So archaea possess certain metabolic similarities with the eukaryotes, but they do not possess nuclei. That's why they are not considered as bacteria, but they are said to be archaea. So we will be studying over here about the archaea. Next we will be studying, that is the word eubacteria. Eubacteria, or you can say true bacteria, are single-celled prokaryotic microorganisms that have a range of characteristics and are found in various conditions throughout the world. All the types of bacteria fall under this title except the archaea, or you can say archaebacteria. So one is archaea that are not bacteria, others are eubacteria that are true bacteria. Next we will be studying, that is gram-negative bacteria. You all are aware what are gram-negative and what are gram-positive bacteria. So there is just a brush up. They are characterized by their cell envelopes, which are composed of a thin peptidoglycan cell wall sandwiched between the inner cytoplasmic cell membrane and bacterial outer membrane. Gram-negative bacteria. Example, you all are knowing E. coli, Pseudomonas, Chlamydia, etc. Right? Why they are termed as gram-negative bacteria? Because they are stained which color? They are stained pink in color. Why? Because they retain the stain safranin. Next is aerobes. What are aerobes? The organisms which compulsorily require oxygen or air for their living, that are termed as aerobes, or the bacteria are known as aerobic bacteria. So aerobic means they are exposed to air or an oxygen, right? Without air or oxygen they cannot survive, so that are aerobes. And the bacteria which require aerobic environment, that is aerobic. Next is anaerobes. Anaerobes means absence of oxygen. So generally those organisms which do not require oxygen or air for their growth are termed as anaerobes. The catabolic metabolism of anaerobic microorganisms reduces a variety of organic and inorganic compounds like CO2, sulfate, nitrate, etc. Anaerobes produce large number of end products of metabolism: acetic acid, lactic acid, ethanol, methane, etc. You have already studied some parts of fermentation where anaerobic bacteria takes place. Next is anaerobic environment. You all are smart enough to understand that the environment without air or oxygen is termed as anaerobic environment, and the organisms which survive in anaerobic environment are termed as anaerobic bacteria. Next terminology which we are going to discuss, that is microaerophilic. Micro means a part, aero means air, and philic or phile, that means loving. So the organisms that requires oxygen but requires environments containing lower level of oxygen. Micro means trace amount, very small amount of oxygen is required for their growth, that are regarded as microaerophiles, that is organisms, and the organisms which survive in microaerophile environment, that is microaerophilic. Next is facultative anaerobes. A facultative anaerobes is an organism that makes ATP by aerobic respiration if oxygen is present, but is capable of switching to fermentation or anaerobic respiration if oxygen is absent. Facultative means it is not compulsory. Oxygen is present, they can grow, they can survive. If it is not present, they have certain mechanisms in their body that they will switch over to an aerobic respiration process. So they are termed as facultative anaerobes. An obligate aerobe, by contrast, can make ATP. See, facultative anaerobe and obligate aerobe. Obligate means compulsory. Aerobe means they require air or oxygen in compulsion manner. So one term is facultative anaerobes and another term is obligate aerobes. By contrast, cannot make ATP in absence of oxygen. So those who require compulsorily oxygen, they cannot produce ATP or they cannot do metabolism. And an obligate anaerobe die in the presence of oxygen. Obligate means compulsory. Aerobe requires air. Obligate anaerobes compulsory, they do not require oxygen. If oxygen is given, they will die. Next is dissimilatory sulfate reduction or and reducers. So dissimilatory sulfate reduction is a form of anaerobic respiration that uses sulfate as the terminal electron acceptor, right? So that are termed as dissimilatory sulfate reduction. And the metabolism which is found in some types of bacteria and archaea, which are often termed as sulfate producing organisms. So these organisms where this type of mechanism, that is dissimilatory sulfate reduction, is done, that are termed as sulfate reducing organisms, or they are termed as sulfate reducers. Next we will be studying that is phototrophic bacteria. Photo, that means life. Photo, that means light. A group of bacteria including green bacteria and purple bacteria whose energy for growth is derived from sunlight and whose carbon is derived from carbon dioxide or organic acid. These type of organisms or bacteria are termed as phototrophic bacteria. Hope you all are understanding. Phototrophic means they are deriving nutrition from the sunlight. Next is gram-positive bacteria, pertaining to the bacteria holding the color of primary stain when treated with the gram stain or gentian violet, right? They are gram-positive. Positive are stained purple in color. So the bacteria which holds the blue or purple stain. Why? Because of their mechanism which we have already discussed in your previous semesters. Because of this, blue crystal violet dyes with bacteria are stained does not wash off of the cell and bacteria appears blue under the microscope. Why? They appears blue or purple, the reason is given, right? Next is endospores. Endospores are tough non-reproductive structures which are dormant. What do you mean by word dormant? Dormant means they are either inactive or not metabolically much active. They are produced by a small number of bacteria from the Firmicute family. Racillicutes, Firmicutes, these all are the families of the Burgess manual, that is classification of various types of microorganisms. So endospores, generally gram-positive bacteria forms endospores. The primary function of most endospores is to ensure the survival of a bacterium through periods of the environmental stress. What is the main function of the endospore? Why they are produced? Endo means inside, exo means outside. So two types of spores are there, endospore and exospore. Endospore means inside the whatever there is there inside the bacterium, it will be hold together for a long duration of time to overcome the environmental stress. Next is Mycobacterium. You all are aware about this word, Mycobacterium tuberculosis, Mycobacterium leprae. Mycobacterium is a wide category of bacteria. Over 190 species are there inside this genus. This includes pathogens which cause severe disease in mammals or animals. These whole things, mycobacterium, regarding mycobacteria, we will be studying in this unit. Then we will be studying about the actinomycetes, that is gram-positive mycelial bacteria. Actinomyces, we all are aware that they form a chalky colony, the earthy smell which is found after the rainfall, that is because of these actinomycetes. And actinomycetes is a whole family where various actinomycetes, streptomycins, etc. are included. Next is extremophiles, as I already discussed in previous slides. Extremophiles are the organisms that have been discovered on the earth that survive in the environments which is not favorable or reasonable for the growth of the common organisms. These extreme environments include intense heat, highly acidic environments, highly basic environments, extreme pressure, extreme cold, etc. So these are termed as extremophiles. This is the list of various reference books which are prescribed in your syllabus. I request you all to go through them and keep reading these reference books. If you are not having hard copy, it's okay. Even you can download it from the net. You can use ebooks and study, because whatever I am going to discuss here will be just a supportive material. Study you have to do, you have to prepare the notes, you have to read extra material from various sources and prepare your notes, so it will be easier for you to write in the examinations. So these reference books are there, go through it, find it out, and if you are having some doubts or query, you can always contact the teachers. So hope you all like this video, like this presentation, and hope you all became just aware what we are going to discuss in this particular unit in your paper number 301. No doubt some of the words are a bit difficult for you to understand, but as and now when we are going to discuss, we will be discussing in detail about each and everything, whatever is described in the syllabus. So study hard, study well, and hope you all are safe at your home. Do not go out unnecessarily without any reasons for roaming. So with this, today I am ending my lecture over here. Thank you all so much.